Okay. Oh, well, I think we can start and people will join after it. Welcome, everyone. It's my pleasure to introduce our speaker, Dr. Nobuto Takeuchi. Nobuto is a theoretical biologist. He did his PhD in Amsterdam with Pauline Hoogeveg on the evolution of RNA. After that, he moved to the US to work with Eugene Kooning on horizontal gene transfer during a postdoc. Later, he returned to Japan, where he's originally from, to do a second postdoc on the evolution of the central dogma. And after that, he moved to New Zealand, where he's been working since 2018. It's all to you, Nobuto.
Thanks, Alex, for inviting me to this to seminar series and giving me opportunity. I'm glad to be able to present our work, and it's a bit pity that I can't have this seminar in person. I would have liked to meet all of you. Okay, so I'm going to talk today about our computational modeling work on the reproductive division of labor. And the conclusion of our research in a nutshell is basically making a prediction that is opposite to what people have often predicted using kin selection, namely that reproductive division of labor evolves only if relatedness is high enough. And we are going to show a conclusion which is opposite, that it evolves if relatedness is low enough. But before getting into that conclusion, let me start with what reproductive division of labor is. So reproductive division of labor happens between reproducers and helpers. So these are different types of individuals forming a group. So within the group, there are two kinds of individuals: reproducers, which transmit genetic information across generations, and helpers, which assist reproducers in transmitting genetic information. However, the helpers cannot transmit genetic information across generations. They might be able to replicate for a few times, but eventually they will not be able to continue their lineage. And example of reproductive division of labor, or RDL for short in this talk, includes eusociality. So the colony of eusocial insects consists of many insect individuals, and some individuals, called queens, transmit genetic information, so they are the reproducers. And the worker bees and ants, workers, they assist queens to transmit this information by performing various tasks that are beneficial for the colony, such as defending against enemies and so forth. And another example of reproductive division of labor at a distinct level of biological hierarchy, namely the cellular level, is the germline-soma distinction in multicellular organisms. So germline cells, the gametes unite and form a germline cells, so they can transmit information. And somatic cells, which derive from a progenitor of germline cells, they do all sorts of somatic tasks to help germline cells to transmit information, but the somatic cells basically die with an individual, so they don't transmit genetic information. Same kind of separation. Now, examples continue. It also occurs, RDL also occurs at organella level. So this is a schematic drawing of a tetrahymena, which is a ciliate, a protozoa. And this organism contains multiple nuclei, and one type of nuclei called micronuclei or nuclei transmit genetic information. So these guys undergo replication, cells replicate. And another kind of nuclei, macronuclei, which derive from micronuclei, they do not transmit information. They don't undergo replication, and instead they express genes, which then perform all sorts of functions that are beneficial for the reproduction at the cellular level. So this is again another example of the distinction between information transmitter, the reproducer, and the information expressor, which is the helper. And Yet another example happens at bacterial level. So this is a simpler cell level. So this is a schematic draw. Well, it's actually a photo of a plant fopper insect and bacterial symbionts living in this organism display distinction between germline cells and somatic cells. So the insect has two kinds of organs that house this bacterial symbiont, and one organ which is possessed only by the female but not by the male insect, has these small cells. These are the bacterial cells living in this organ, and they are transmissible to next generation. So basically, the mother transmit these bacterial cells to her eggs. And these other organs, which are possessed by both male and female insects, the bacterial cells in it are humongously enlarged, as you can see in a different scale of cells, and they produce metabolites and they can't transmit genetic information. And yet another example of reproductive division of labor happens at the molecular level. So cell consists of multiple molecules, and they're divided into two kinds of molecules, broadly speaking. One kind is called genes that transmit genetic information, and enzymes, which are derived from genes, but they themselves don't transmit information, and instead they provide chemical catalysis. So the information transfers from genes to genes or genes to enzymes, but never from enzymes to genes. And this is also called the central dogma of molecular biology. Now you might think that I'm repetitive, and if that's the case, then this is exactly what I wanted to say. So reproductive division of labor evolved repeatedly across entire different biological scales, and almost at every conceivable level of biological individual, reproductive division of labor has been observed. And so this repeated evolution of RDL begs a question. So why does RDL evolve so repeatedly across different levels? Is there any general principle behind this? And the previous authors have put forward what we term the efficiency hypothesis, which in this slide I call it classical hypothesis for simplicity. So the previous hypothesis, the existing hypothesis, posits that reproductive division of labor evolves because it increases group level production efficiency. And here the group level production efficiency refers to fitness per unit resource investment. So that's schematically shown here. So on the left-hand side, the individuals are forming a cooperative group, so they are forming a collective, and don't divide labor. That has a low efficiency at the group level production. And if they divide the labor, then efficiency increases, so they can produce more. Now, this hypothesis, which basically says RD evolves because RD is good, needs an assumption, namely that the return on investment is accelerating. So suppose these individuals have to produce some public good that is required for reproduction. And if they invest more in public good production, the return will of course increase. And if this return is accelerating, that is to say the unit investment brings more return if you invest more, in that situation, it actually is better to divide the labor so that subset of individuals invest everything into public good production, and the remaining subset will invest everything to self-reproduction, and that maximizes the overall growth rate of a group. So that's the classic hypothesis. Now, this is formulated in quite a general terminology, and therefore it leaves the question like why exactly this efficiency increase happens in actual biological systems. And to that question, the different authors have different answers for different systems. So for example, in Volvox, which is a green alga, a germline-soma separation happens in this organism. And the authors, previous authors said that the reproductive division of labor in this organism is good in terms of hydrodynamic body design. It allows hydrodynamic body design and therefore increases locomotive efficiency. So germline doesn't contribute to swimming, and somatic cells contribute to swimming, and that somehow increases efficiency of the locomotion. Another author said that the gene-enzyme reproductive division or whatever that happens in a cellular within a cell increases the efficiency of or replication overall by optimizing catalytic activity of enzymes and hydrolytic resistance of genes. So in RNA world, the same molecule has to be both catalyst and template. So this is before the distinction between genes and enzymes. And once this distinction evolves, the gene is now free to, they don't have to have catalytic activity, so they can maximize their resistance against hydrolysis. And similar is true for enzymes, but for the catalysis, and that increases overall productivity. Now, maybe there is some common theme behind this, but at least the way it is phrased, there doesn't seem to be any sort of general applicable rule, but more rather these mechanisms seem system specific. And to that, in this talk, I want to propose this new hypothesis, which we call cheetah hypothesis, as a unifying mechanism to explain reproductive division of labor at different levels of biological hierarchy. But to explain this hypothesis, I need to first explain what cheetahs are. So cheetah. Sorry, I muted myself, sorry. So suppose you have a group of cooperators on the left. So each individual has to either replicate or produce public good. And cheetahs are the individuals that don't produce public good and dedicate all its resource to self-reproduction. And such cheetahs can emerge by mutation, and this is experimentally known that there are many cheetahs in different systems. And these cheetahs pose an issue for group level collective living. And so that is because when the cheetahs arise, so this is a group, and this is time progresses from left to right. And by mutation, cheetah appears. And suppose all individuals, they have to spend a fraction of the resource in self-reproduction, and fraction of resource in producing public good and help each other. And suppose cheetah arises, and this cheetah doesn't invest anything in public good production and divert all its resource in its self-reproduction. That means that this fitness of this cheating individual within this collective will increase by this diversion of resource, and therefore it will spread within a group. But if a group contains only cheaters, then there is no one producing public good. So that group will not perform very well. So this is the problem of cheaters. If cheaters arise, they can spread and decrease group level fitness. And our cheetah hypothesis posits that the reproductive division of labor inhibits the evolution of cheetahs. And this is how it works. So on the top, we have these individuals doing both replication and public good production. On the bottom, we have a system with reproductive division of labor, where this orange reproducer, they focus on producing more and more individuals. And the blue ones, these are the helpers, they are dedicated to production of public good, and they are not capable of transmitting genetic information, at least across indefinite number of generations. And in this situation, the cheetahs could still arise by mutation, but because the majority, if the majority of individuals are helpers, then cheetahs that are arising in the helper population will not be able to spread because the information, genetic information is not spread. So cheetahs, if cheetahs were to evolve, they must appear in the reproducer population, but because the reproducer population is small, or if it is small per group, then basically the number of individuals subject to mutation, the relevant mutation that generates cheetahs will decrease. So there will be less chance of cheetahs evolving. Now, other way of saying this is that there is a kind of effective population bottleneck happening because of this reproducer helper distinction, where the reproducers are the only genetic information transmitter, and thereby this bottleneck increases genetic homogeneity of individuals within the group, which is same thing as saying that relatedness within the group increases, and hence it's harder for cheaters to invade. So this is our hypothesis. Reproductive division of labor has this advantage of increasing relatedness and thereby inhibiting the evolution of cheetahs. Now, this cheetah hypothesis has an advantage over classic hypothesis in that it is applicable to any systems where cheetahs can prosper. And in fact, cheetahs is known to occur across many systems and scales. So, eusocial insect has cheating workers, egg-laying workers. Multicellular organisms has cancer cells. They are cheating cells. And in the cellular level, experimental evolution has shown that there are lots of opportunities for selfish template molecules to arise in the molecular system. They are the molecular cheetahs. So, so because of that, cheetah hypothesis is applicable to various systems where cheetahs can arise. And another interesting thing about cheetah hypothesis is its prediction. It makes interesting prediction. So, our hypothesis posits that the RDL, the reproductive division of labor, is beneficial against cheating, right? Well, this means that the corollary of that is that RDL evolves if risk of cheetah evolution is high. If there is no risk of cheetah evolution, then there is no benefit of RDL. That's our hypothesis. Now, this makes an interesting prediction because when is the risk of cheetah evolution high? And that is when relatedness is low. So that, sorry. That's right, yes. So when the risk is high, when relatedness is low. That means that the RDL is predicted to evolve if relatedness is low. So that is schematically shown in this diagram here. So if the relatedness is high, so that means that there is not much genetic heterogeneity within a collective, then cheetahs are not a problem. So therefore RDL will not evolve. But if relatedness is sufficiently low, genetic heterogeneity sufficiently high, then there is a kind of evolutionary natural selection incentive for reproductive division of labor to evolve. And that is in contrast to a classic prediction made by kin selection theory, which posits that the reproductive division of labor evolves if relatedness is high. And I think many of you have learned in undergraduate evolutionary biology course that eusocial insect evolves because of haplodiploidy, which is actually debated in kin selection theory. But the point of this statement is that reproductive division of labor evolves if relatedness is high. This is a textbook notion, and our cheetah hypothesis makes a prediction that is in diametric opposition to that textbook statement. So we want to test this idea that reproductive division of labor evolves if relatedness is sufficiently low. So we made the simplest possible model that we could come up with. And I will explain this model in terms of cells grouped into multicellular organisms, just because that it's just a terminology easy to understand for biologists, including myself. So the model consists of individual cells, the population of cells that are grouped into a population of organisms. So this big circle is organism. And organism can grow as cells divide. And when the number of cells reach certain threshold, organism can split into two. And the cells within the organism are randomly split between two organisms. And cells can also die, which then can cause a shrinkage of an organism. And if there are no cells in organism, then organism also dies. And there are two kinds of cell types, two cell types assumed in this model, which we call P and Q. And they can go back and forth. The cells can change its state or type with some transition probability denoted by dP or dQ. And each cell has to decide how much fraction of resource it has on zomatic investment. So Kp is a fraction of resource that this cell invests in zomatic function if a cell is in state P. Okay, and 1 minus Kp is the remaining fraction, which is invested to self-reproduction of this cell. So this is a little difficult part to understand. So just to please pay attention. So the cell can take two cell state, okay, P or Q. They can transit. If a cell is in state P, then it invests fraction Kp in zomatic investment. But if that cell is in state Q, then it invests KQ fraction into somatic investment, okay? So every individual cell in the model has this basically 4 parameters assigned, KP, KQ, DP, DQ. And depending on whether a cell is in state P or state Q, it expresses, so to speak, a different set of parameters. So if the cell is in state P, fraction Kp is invested in somatic investment, and with probability of dp, it becomes q. And these parameters evolve by undergoing mutational selection as a cell replicates. And the resource is computed between organisms. So there is a total population size that is bounded above. And fitness, which is basically the reproduction rate of a cell, is defined by this formula, which I will explain. So 1 minus k, so this x is either p or q, but 1 minus k is the fraction here that the cell invests in its own private reproduction. And this angular bracket k is the amount of somatic investment averaged over all cells within an organism to which the particular cell belongs to. So this is basically how much cooperation going on in the organism in which cell belongs. And 1 minus k is basically how much you invest in your self-reproduction, okay? And the important thing to note is that this x is just p or q. It's not a number. So this is a linear to 1 minus k, and it's also linear to k. So it's a linear in both investment in public good production and self-reproduction. And that means that in this model, there is no benefit whatsoever in dividing the labor. Okay, so dividing labor or not doesn't have any influence on overall growth rate of cells. Now, I have explained the model. Do you have any questions so far? Please feel free to just jump in to ask questions, and I will explain more details of model or introductions or whatever, if you have any questions.
I was wondering actually why is the P and the Q similar across simulations or in terms of magnitude? Or is that how?
That's a very good question. Sorry, yeah, I did not say that. So indeed, so at time 0, I set dp2 and dq to 0.5. So probability 0.5 of transiting to q when p replicates, then with probability dp, it becomes q. Probability 1 minus dp, it stays p. And at the beginning of the simulation, I set dp to dq to 0.5, which means that the cell state transition is completely random at time 0. So there is no bias towards becoming p, bias towards becoming q. And then what I will do is I will let these parameters mutate. So every individual cell has its own private set of parameters, which are inherited when the cell replicates and also undergo mutation. That means that the distribution of DPDQ parameters in the population of cell can change the location as well as, of course, variance too, so that there may be a skew in the way P or Q differentiate into each other. And the same thing is true for KP and KQ. The beginning of the simulation, I set KP and KQ to 0.5, which is the maximum you can achieve. As you can see from this equation, when K is 0.5, this is takes a maximum value. So at the time 0, I assume that there is no difference between P and Q. So Kp is same as Kq, dP is same as dq, and so there is no functional differentiation between P and Q. And my question then is whether evolution will then generate symmetry breaking between these parameters. Did I answer your question?
Yeah, that's exactly what I meant.
Okay, good. Any other questions? Please feel free. You can also write in the chat if you want to. Okay, if not, then I'll just move on to the result. So here is the result. So what I did, I ran this simulation, this model for different parameter values, focusing on two kinds of parameter. One is mutation rate parameter, so that's just probability of how, whether traits such as DP and DK and so forth will change when cell replicates. And another parameter is a split threshold V, and that is the number of cells above which organisms split. So V controls population size of cells per group, per organism, and M is mutation rate. So obviously, population size and mutation rate are important parameters of evolutionary dynamics, and that's why I'm varying these two parameters in the two-dimensional space. And I see a different outcome of simulation depending on these two parameters. And I will focus on these two regions that are important for me to make conclusion. So one region, this cyan region, that's a low mutation rate and low number of cells per group. This is where the cells remain undifferentiated. So these colors, DP, DQ, and public good investment, are corresponding between these two pictures. And the x-axis is time, and the y-axis is average value of these traits over all cells in the system. And as you can see that Kp and Kq take same value, so there is no differentiation in terms of how much somatic functions they perform. And there's a wide fluctuation in the rate at which P differentiates to Q and vice versa. The time is, by the way, log scale. So this is different time scale depending on the location of x-axis. And when these two parameters, mutation rate and split threshold, increase, then something interesting happens. And this is a symmetry breaking. So, these parameters evolve different values. And at the end of this simulation, after reaching equilibrium, I see that these two parameters are positive and these two parameters are zero. And if you map that into this schematic drawing, what you see is that P differentiates into Q, but Q does not differentiate into P. As you can see that this light orange is zero. And Q provides public good, and P doesn't. And this means that P is the germline cell and Q is the somatic cell. Because the differentiation is one directional from P to Q, even though Q replicates, it turns out that Q, the lineage of Q, if you trace back the lineage of Q, it quickly reaches the P. So basically all Q cells in the system are recent derivative of P, and that's why Q is not transmitting information. So this is the establishment of the reproductive division of labor. So at least our model shows that without assuming any nonlinear return in dividing labor, reproductive division of labor evolves and it's stable. And our hypothesis is that that's because it increases relatedness. So let's see whether that's true. So what's shown here is the frequency fraction between 0 and 1 of P cells in a group, averaged over all groups, depending on the parameter. So when we are in a parameter region where reproductive division of labor does not evolve, then P and Q frequencies are basically 50-50, as you'd expect. But if the reproductive division of labor evolves, then the germline cells are always in minority. Now you might wonder, like, well, does P have to be always germline? Well, the answer is no. Actually, the chance is 50-50. P or Q become germline cell, but just for the sake of presentation, I'm always assuming that P is a germline cell, but that's not the case in actual simulation because this is a symmetry breaking. You have this chance, 50-50 chance of which way you break the symmetry. Now, the important bit here is that population size of the cells that transmit genetic information is minority. In minority, it's very small. And because of that, there is a population bottleneck effect. And because of that, the relatedness, which is plotted here, increases as reproductive division of labor evolves. And in conjunction, the reproduction rate averaged over all organisms will also increase. So this is all fitting, agreeing with the hypothesis which posits that the reproductive division of labor evolves because it increases relatedness and hence the average productivity of a cell because it stabilizes cooperation between individuals. So the next question is then, so the hypothesis predicted that the reproductive division of labor should evolve when relatedness is low, okay? When the risk of cheetah evolution is high, then reproductive division of labor should evolve. That was the hypothesis, so the prediction of the hypothesis. So what we want to know next is whether this magenta region has indeed lower relatedness than a cyan region. Now, to address this question, we have to allow for the fact that relatedness is actually not constant throughout time. Okay, it depends on the evolution of reproductive division of labor, because the hypothesis assumes that reproductive division of labor increases relatedness. So to answer this question, whether reproductive division of labor evolves if relatedness is low, we must measure relatedness just before the evolution of reproductive division of labor if reproductive division of labor evolves. And that relatedness we call baseline relatedness. Okay, so this is the relatedness before reproductive division of labor. And after reproductive division of labor, we call it post-RDL relatedness. And of course, if the simulation doesn't show evolution of reproductive division of labor, then baseline and post-RDL are indistinguishable. And the result is shown here. So the x-axis is just a rank. So basically all these simulations come from these simulations that are either in cyan or magenta, and they are ranked by baseline relatedness, okay, from 1 to 60. And this red part, this is the post-RDL relatedness, okay? So what this means is that the reproductive division of labor evolves in these bottom ranking simulations for which the baseline relatedness is lower. Okay, so this agrees with the prediction of the hypothesis that reproductive division of labor evolves if relatedness is sufficiently low. But because relatedness increases, reproductive division of labor increases relatedness, so reproductive division of labor and high relatedness coincide in our model, which is in agreement with kin selection theory. But the difference is that kin selection theory has been used to say that high relatedness is the cause of the reproductive division of labor. Here, not disagreeing with kin selection theory, we are saying that high relatedness is a consequence rather than cause of the reproductive division of labor. Now, this is the main result, and because this is the theory of living matter, I thought maybe just good to mention, but I will not go into details of a mathematical analysis of all this simulation. So if we set this cell differentiation parameter to 0.5, we fix it, then the system becomes two-dimensional system. We have only Kp and Kq, and that then becomes amenable to mathematical analysis, and we can use simple matrix population model to describe this growth of this cell population within the group with fitness defined just like we did for computational model. And I will skip over the details. However, we're using the equation, so-called price equation, which is often used in kin selection theory. We can derive mathematically that there is a mathematical positive feedback mechanism, which says that if public good, so zomatic function, zomatic investment of one cell type is slightly greater, tiny bit greater than the other cell type, then this difference expands, and therefore there will be a symmetry breaking. So this is a mathematical consequence of this model. But I will skip the details here for the sake of just to keep it simple. Okay, so let me now move on to discussion. So our hypothesis, sorry for repeating, says that RDL evolves as a cheetah prediction for low relatedness, okay? And one advantage is that hypothesis is general. It is applicable to any biological systems where cheetahs can prosper. And it is known that many systems cheetahs prosper. So that's why the hypothesis is general. But advantage of that, of this hypothesis is not only that it is general, but also somewhat, it sounds like oxymoron, but it's also specific. That is to say that this previous hypothesis, which we call the efficiency hypothesis, okay, the nonlinear accelerating return and hence RDA is good and therefore it evolves hypothesis, has an issue in that when we say division of labor is good for group, it can be any sort of division of labor between any sort of work, any sort of labor. Okay, it doesn't have to be between reproductive labor and non-reproductive labor. It can be between any arbitrary pair of labor, as far as hypothesis is concerned. Okay, however, a scientist called Simpson, this person looked at all sorts of division of labor that happened in a system that consists of collective of individuals. So that is to say, a system that has undergone a major transition, specifically the fraternal major transition, if you know. For those independent, phylogenetically independent cases of major transition, and this person looked at, like, does the division of labor evolve? If it evolves, is it reproductive or non-reproductive? It turns out that in more than 100 separate origins of the division of labor happening in the system that has undergone major transition, either only reproductive division of labor evolves and non-reproductive division of labor evolve, or reproductive division of labor precedes non-reproductive division of labor. So let me give you an example. So for example, in eusocial insect, you probably know worker bees. There are different kinds of worker bees, like soldier bees or harvesting bees. I don't know if it's that's the correct terminology, but bees, not bees, maybe ants. Ants, the correct food, and ants that fight against enemies. There is a division of labor within workers. That is non-reproductive division of labor. Then the queen worker division, that is the reproductive division of labor. And it's well known that the reproductive division of labor evolves first, and then non-reproductive division of labor evolves, at least in eusocial insects. Now that's true also for multicellular organisms. Now this means that the patterns in the nature say, basically, very clearly, that reproductive division of labor has precedence somehow relative to non-reproductive division of labor. Now, to explain that, the classic hypothesis, which says division of labor is good because it increases efficiency, has to add an extra assumption or has to come up with a separate explanation as to why reproductive division of labor is more conducive to the increase in efficiency than non-reproductive division of labor. Now, in contrast, our hypothesis is specifically applicable to reproductive division of labor, and therefore it does not have to explain. It already explains the precedence of reproductive division of labor. So that is the specificity advantage of our hypothesis relative to existing hypothesis. Okay, so that's all I need to say now. Let me acknowledge. So Dominic Devlin, Austin Gunley are my collaborators on this project, and also funding from Oakland. Thanks. Any questions? Sorry, I forgot to ask questions during my talk after model.
Thank you so much, Nobuto.
Welcome.
I have a question actually to start things.
Yeah.
Have you identified any cheater phenotypes in your model that would then motivate the evolution of reproductive
Yes.
division of labor?
Yeah. Yes. It does. So when the simulation starts, there is no difference between P and Q, as I said, and cheating behavior that does evolve in this situation is those cells that don't invest in somatic investment. So those are the cells that reduce Kp and Kq and commensurately increases Kp and 1 minus Kp and 1 minus Kq. So those cells living in the same group as the cells that do provide somatic investment, these cheetah cells will spread within the group because they have faster reproductive ability. Now, after the division of labor, what would be the cheetahs in this system? Now, cheetahs would then be not the queues that don't provide public good. I mean, yes, you could call them cheetahs, but those will not spread anyway because Q cells are not transmitting information. So in this system, the cheetah cells would be a P cells that don't differentiate into Q cells. Okay, so P cells, they are germline cells, they're proliferating, but they also have to produce Q cells. Otherwise, no one will produce public good. So Q has to sometimes sacrifice its reproduction to generate Q. That is the actually public good production, and cheating will be those P cells that don't do this differentiation into Q. So cheetahs can arise. The difference between system with and without RDL is that this system, the population size of P is small, as shown here. And because population size is small, the target of mutation is small, so less chance of cheetahs arising. Did I answer your question? Did I don't hear anyone saying anything.
People can also type it in the chat and I will read the question if that's easier.
Yeah,
But
sure, yes, if you
while we
have
wait,
question.
another question that I was thinking of actually is
Mm-hmm.
in this chart that you have in the middle, it's very
Mm-hmm.
clear that the split threshold for the organisms and also the mutation rate have a big impact in which strategy evolves, right? Whether differentiation happens or not.
Yes. Yeah.
Why is that? I just
Yeah,
couldn't quite.
yeah, it's not there is a simple reason for that. So when the population size is small, then more random genetic drift occurs, right? And if the population size is large, then there is less genetic drift. So random genetic drift removes any genetic variation in the population. So that means that if you decrease split threshold, which is to decrease population size of cells per group, then you're increasing random genetic drift and removing genetic variation, okay? Mutation rate, if you decrease mutation rate, well, that's of course reduces genetic variation. So that means that reproductive division of labor evolves if genetic variation within a group is large enough, which is basically the same as saying that relatedness has to be small. Okay, relatedness refers to how closely genetically individuals are related within groups relative to between groups. And that's the relatedness. So that means that if you have uh, higher heterogeneity, genetic heterogeneity, then relatedness is lower, or higher genetic homogeneity, relatedness is higher. So our hypothesis says that reproductive division of labor is beneficial against cheating, but that is beneficial only if cheating is an issue, and when is cheating an issue? Well, that's when relatedness is low, and that is this region.
Perfect. Yeah, that makes sense. Thank you.
Welcome. Okay, so there's a question. Yeah, welcome. Did you ever find that RDA was lost? Oh, yes, yes, precisely. This triangle region is the regions where RDL evolves and is lost. And the reason is, let me just read on. For example, if the probability of increase has to be larger. Yeah, so the reason is that as we get closer to this region, then the relatedness, the baseline relatedness is really, really low. And so reproductive division of labor evolves to increase relatedness, but of course, increase is finite. Now, to increase really, really relatedness, what they try to do, what the organism try to do, is to decrease population size of P cells a lot, to have a really, really tight bottleneck. But then once in a while they lose P cells, they lose germline cells, and then system just collapses. So I think that this instability is due to the simplicity of our model. That is that when organisms split, cells are randomly assorted. And that's not always true. It's true in very primitive organisms, but that's not true for more sophisticated organisms. So because of this simplicity, there is instability. So the reason is because P cell frequency is so low. Did I answer your question, Zoe? Yeah, welcome. Any questions? Any remark? Anything? You can feel free to ask any questions, or even not even questions, even remarks, or even like any random comments you want to make. Can the transition lines be estimated analytically? Yes and no. Probably I can't. So we made a very simplified model without any P and Q distinction. It's just P, okay, just P. And in that system, without Q, just P, the evolution either increases public good production to a good level, or cheetahs invade and public good production will tank to zero. And so there is a kind of transition. Okay, so cheetah invading, taking over, or cooperation maintained. And that transition line is equivalent, almost qualitatively, but not quantitatively equivalent to this kind of transition, because that is a transition line where relatedness is too low to maintain public good production. For that, we were able to estimate analytically, and we published that in genetics, I think. So if you can look up my Google Scholar list, genetics paper. But the derivation does make an assumption. So if it's probably you are mathematical biologist, maybe I can say. So we solve price equation, and price equation is in terms of a first moment, second moment, third moment, so forth. And so we have to do the moment closure, and we close the moment by saying that the cubic moment is certain power of second moment, and because the physical unit is the same if you take proper power. So that's, assumption. Okay, so but the earlier transition is not the cheetahs taking over rather than the number shrink. So it's a different type of transition, right? I don't think so. I think the earlier transition is qualitatively same as cheetah taking over. That is to say that if the risk of cheetah is high enough, then RDLL is beneficial. That's what I said. So this line is a line saying that beyond this, cheetah is really bad. Okay, that's why I'm saying it's qualitatively saying. Now, then why is there a particular line there that the cheetah is changing continuously? Why is there a particular line there? And I think that is that when they have reproductive division of labor, although I said that the return on investment is linear, and therefore it doesn't matter whether they divide labor or not, that's true if number of cells is infinite. But if number of cells is finite in this system, and if the cell number of cells is finite, then there will be fluctuation in the frequency of P and Q, and that will disturb the composition of P and Q. So there is optimal composition of P and Q in this system, and that is disturbed. So actually, reproductive division of labor, if there is no cheetah involved, is actually a bad strategy compared to non-division of labor. So this is actually the preferred strategy. And that means that advantage of this must be outcome, like outweighed by the advantage of cheetah prevention. So there is a threshold, like there is some kind of baseline advantage for not dividing labor, and advantage of dividing labor has to exceed that. And that advantage comes from the fact that cheetahs are risk, poses a sufficient risk. And then when this risk takes over, reproductive division of labor evolves. Now, if reproductive division of labor cannot evolve, then cheetah takes over, and then public good production is not maintained. So that's why I'm saying this is qualitatively the same thing. Okay, thank you. Yeah, so the next question is eusocial insects. Can we make a model of eusocial insects and show the same thing? That's the next question.
I guess the model is quite agnostic to.
Yes, that is a good point. And somehow the eusocial insects or eusocial or animals, when you read eusocial animals, like what is eusociality, it defines not just by reproductive division of labor, but by the fact that children stay with their parents, which is another way of saying that they form a collective group. So when we say eusociality, it seems like it includes both formation of group and then reproductive division of labor. And that to me, in my current thinking, implies that the individuals should have ability to just leave the group, go to another group, or maybe form a own group or something like that. And that kind of process, how does that work out is one question. The other question is that in this reproductive, in my model, which I framed as multicellular organisms, the cells replicate multiple times within the group. And if they don't divide labor, then all cells replicate multiple times within the group. Okay, on average, of course, they replicate once and then become twice and they split. So on average, once, but some cells replicate more than the other cells. And so the number of replication per cell within the collective is on average one, but there is a variance, okay? Now, for eusocial insect, there is not this kind of, you know, sometimes eusocial insect has these queens replicate only twice or something like that. I forgot the specific terminology for that kind of thing. So how does this kind of things work out? In my model, I talk about mutation. But is mutation rate large enough in eusocial insect? Or should I talk about epigenetic things? Or is it just from a theoretical point of view, same? These kind of things I'm still thinking. I don't know. What do you guys think? Do you think this model already shows that eusociality is explained and I don't have to make a eusocial insect model?
I think you are right, indeed, that uh, there is an aspect to the group clonality that is kind of implied in the model, right?
Indeed, indeed, indeed. Eusocial
Um,
insects are diploid and they are sexual. Some insects are like aphids are sexual, asexual, but yeah, many are sexual. So how does the non-clonality?
and also in the sense that you, for example, the model that I work with, we model multicellularity as an aggregative process, right? So there we have
Mm,
groups coming together. So
mm.
groups that are hybrids of groups that were once clonal. And so I think
Oh, I
kind
see.
of
Yeah,
this
yeah,
sort
yeah,
of feature
that,
might
yeah.
be necessary to model social insects, or at least it's qualitatively kind of equivalent with the ability to leave and join another group, right?
Mm, indeed, yeah. It's sort of like, yeah, indeed. It's a question of like, does this process that we are coming up with, is this really robust enough in a specific context with sociality? Just, I guess that's probably a necessary thing to convince people who are working in new sociality. Or maybe it doesn't work. No.
There's another question in the
Yeah.
chat.
So would
Yeah.
you be able to comment how you'd imagine this playing out in RNA? Do I understand correctly that since there is no linkage between cells, which could mean usually RNA cells, it is, yeah. So yes. So we also made a protocell model, protocell version of this model. And so P and Q are replicators, and they have catalytic activity and catalysis is provided to template, and then template is replicated or transcribed. So it's a model that is rather more specifically geared towards kind of RNA-like situation, where we start with a system where molecule provides both catalysis and function as template, just like RNA world. And then we show that molecules will split into catalysts and templates, so establishing gene enzyme distinction. So it is basically what we are saying is the reproductive division of labor is a way to achieve stochastic correction. It's enhancing the effect of stochastic correction, okay? By decreasing the part of population that is transmitting genetic information, okay? So not all molecules transmit genetic information, only part does. And that part has to be small enough to prevent cheaters. And that can be achieved by dividing labor between the information transmitters, templates, information utilizers, catalysts. So it is a kind of like, yeah, mechanism of creating a stochastic correction. If stochastic correction is interpreted as increase of relatedness, but I think Maynard Smith, Satomari doesn't quite agree with that. I think if I remember his comment in some of the conferences correctly, I think he was saying something more than, there is something more than that. But if we interpret stochastic correction as increase of relatedness, then yes, that we can say it this way.
Thank you.
Welcome.
Good. I think if we don't have any more questions, I'd like to thank you
Good.
again, Nobuto, for coming. That was a really nice talk.
Very
Yeah,
welcome.
thank you also everyone for joining.